historical scientific fact number one: contagion. Take some sick people, expose them to well people. The well people are supposed to get sick. That's what we're told. These things, viruses, germs, fly through the air. They're deadly. Sneezing, breathing, coughing, being within 6 feet of someone who has this deadly thing, supposedly we can catch it and we're going to get sick and die. That's the story. They've tried to prove this hundreds of times. All of them have failed. It's never been demonstrated that contagion is a real phenomenon. In fact, exactly the opposite. It's been disproven. There's this book, Can You Catch a Cold?, where Daniel Roides goes over hundreds of these studies and explains how it's never been shown to be a real thing. Scientific historical fact number two: we're told that these viruses are found, discovered. They do science and prove there's a thing and prove that it's dangerous. That's not at all what they do. And so the steps that virologists claim they do is they take a sample from a human being who's sick. They centrifuge it, which is, separate, filter it, try to get out unwanted things, so there's just the viral or the germs in there, supposedly. Then they culture it. They put it on a cell culture of monkey kidney cells with antibiotics, bovine calf serum, other substances. The point is that it's a mixture of stuff. They don't know what's in there. They didn't separate out this tiny particle all by itself. That's not what they do. So they put it in the cell culture and they let it ferment, culture, or die, break down. They reduce the nutrients which feeds the culture breaks down, it decays. Then they claim that the decay, the dying of the culture is proof there's a virus in there. The scientific term they came up with to describe this is called CPE, cytopathic effect. And the problem with this is that when they do a scientific control, which means they do all the same steps without a human sample, they get the same results, which basically disproves the whole thing, or it proves that the experiment, the steps itself, caused the cytopathic effect. No virus needed, no human sample needed. And even the guy who came up with it, Enders, I think it was measles, he said the same thing. He said, when we didn't add the heavy metals and did the staining and all this, we got the same results. And another microbiologist, Stefan Lanka, has done controls with just yeast and with other materials, and he got the same results. And so that method has proven flawed, yet they still use it. And so that's why in this so-called modern age, they try to steer us or push us towards the more technical stuff, which is like the PCR test, antigen test, genomics, or sequencing, to try to say that, we don't need to do those other experiments. We have the genome, we found it, and it's de novo. We know it's a virus because we have the sequence and we see it from one person to another. The problem with all of these is they assume there's a virus in there to begin with. So there's no gold standard. For example, in a pregnancy test, the gold standard would be you give the test to a woman. 10 months later, either you have a baby or you don't. The baby is the gold standard proof of whether the test works or not. There's nothing like that for COVID. There's no specific thing you can point to that says only this virus has this set of diseases or circumstances. There's a lot of overlap with the symptoms. Coughing, sneezing, loss of taste, smell, these all exist in other ailments, and they existed before COVID, before 2019. The other problem with all these, they don't do controls. By that I mean, we've all heard of placebo and nocebo. And the idea of a placebo effect is you do a random double blind so that the people don't know if they're getting the treatment or a saltwater pill or a sugar pill. With these PCR, antigen, genomic assembly or sequencing, you send these labs 5 samples. Let's say one's lung cancer patient, one person has pneumonia, one person is declared to have COVID, somebody else is declared to have the flu, somebody else is said to have a cold, maybe 6 samples, and a healthy person. And you blindly label these vials of, it's supposed to have this stuff in there. You send it off to the lab and you say, okay, tell us which one's which. And they should be able to do that with 100% accuracy. And then you repeat it. You just send it to other labs with different samples. And they should be able to tell you every single time which one's which. That's science. That's how you prove that this thing is its own entity. Even if all that were somehow proven, they still have to do the most basic thing, which is contagion. Like they say this thing flies through the air and makes us all sick and is deadly contagious. Well, prove it. Take rats or guinea pigs, that word came, guinea pig, being a guinea pig, it came from somewhere. So give it to the guinea pigs. They should get sick. The ones that don't get it shouldn't get sick. Put sick guinea pigs in a room with healthy guinea pigs. The healthy guinea pigs should get sick. That's the story. That's the narrative. If you can't prove that, then it's a BS story and you're just making stuff up. Entire books like this one have been written. Virus Mania, the final pandemic. And so virology has all kinds of problems. And then if we move on to the science of vaccinology, well, first off, it's like there's no proof that these viruses exist and actually do these things. They can't tell one from the other. So how are they going to make a vaccine for something they can't find, they can't prove exists? Like, how do you do that? How do you make a unicorn repellent if you don't have a unicorn, or you don't even know what a unicorn looks like? You're just making it up. How do you do that? Like, logically, what's the how? And so that brings us to the science of so-called vaccines. And the way they make a vaccine is the same way they do the cell culture. In other words, they take a sample, the kidney, monkey kidney, the bovine calf serum, the antibiotics, all these things that they put in there. Sometimes they add formaldehyde, sometimes they add aluminum, sometimes they add mercury. That's what's in a vaccine. That's it. That is the vaccine. They took a cell culture muck soup and then they declared this is the cure. You inject yourself with this and it'll stop these things that we've never found, that we don't know what they look like, and we're just making it up. I mean, for me, I'd want more proof than that. I'd want actual tests. So that brings us to, have they ever safety tested any of these vaccines? And the answer is no. Long-term, double-blind, comparing vaccinated to unvaccinated over the course of 10, 20, 30 years. They haven't done it, or if they have done it, they haven't published it. There have been some doctors who have looked at their own patient data, and they found that the vaccinated kids were sicker than the unvaccinated, or the unvaccinated kids are healthier. Where's a long-term test? The CDC, NIH, they probably have this data. Why haven't they published it? Where is it? Like a full vaccine scheduled group of people to fully unvaccinated. Those populations are out there. They exist. Like, why haven't they done those tests to see what the long-term outcome is? What's kind of shady, or super shady, is when pharma supposedly does vaccine safety tests. Often they compare one vaccine to another. In other words, they invent a new vaccine and they do their so-called safety tests by comparing an old vaccine to the new vaccine. And as long as there aren't more health adverse effects or more dangerous outcomes, more deaths, injuries, they say, oh, it's safe. But they're not actually comparing it to nothing, to a placebo, to an inert thing that doesn't harm people. They're saying, this punch in the face is just as safe as this other punch to the face, instead of saying, well, where's the people you didn't punch in the face? That would be a logical test. Like, you test the thing against something that's nothing. But they haven't done that. Like, if you actually read these so-called safety studies, it's always against some other drug or some other product. So how is that scientific? It's not, in my opinion. So any questions, please leave them in the description or the comments, and I challenge anyone, prove me wrong. Myself and all these doctors and scientists have been looking for years. No one can prove us wrong. No one can show the evidence. Oh, here's where we found a virus. Here's the proof of contagion. Here's the controlled double-blind studies that prove that vaccines are safe and effective. It doesn't exist. It's not out there. If it was, they would have trotted it out a long time ago. Why? Because it would prove their narrative correct. They would make trillions of dollars if they had the proof. There wouldn't be any vaccine hesitancy if they had actual science. I'd do it. I'd get it if the science was there, but it's not. And so I challenge you. If you think I'm lying or full of it, look into it. Find the science. Show it to us. We've been begging people for years. Where's the evidence? No one can find it. AKA, it doesn't exist. So I hope that helps. Hope everyone's well, and thank you so much